Okay, so this video will be an introduction to the human body tissues. And remember, our body is made of lots of specialized cells that all have specific functions. And when these cells come together, they can form a tissue. So by definition, a tissue is basically a group of cells that are similar in structure and then perform a common or related function. And the study of tissues is called histology. In fact, the root histo means tissue. And there's four basic tissue types in the body. We have epithelial, connective, muscular, and nervous. However, there are subcategories of each that we'll get into later in this chapter. So to summarize the different tissue types of our body, remember nervous tissue is what includes your brain, spinal cord, and nerves that are involved with internal communication. Muscular tissue is involved with contraction to cause movement. So you'd find that there's skeletal muscle tissue attached to your bones. There is cardiac muscle tissue that makes up your heart to help pump blood. And then we have smooth muscle tissue that you'd find along your airways and your digestive tract, as well as your genitourinary tracts. And this types of smooth muscles involved with transportation of materials. Now, the epithelial tissues are those ones that cover and line spaces. So if you think about, like, where's a space? Well, you'd find spaces along your respiratory tract. You'd find spaces along the inside of your digestive tract, or even your genitourinary tracts. Even your blood vessels have spaces within them. In fact, all of these spaces, their walls, are lined with an epithelium. And the function of this epithelium is to form boundaries for different environments, like between your tissues and blood, or between the inside of your stomach and the stomach wall. And these are involved with protection, secretion, absorption, and filtration. And this also includes skin. But I'm not saying that skin has all of these functions. You know, like, we don't think of skin as being an absorptive organ, but some epithelial tissues are better at absorption than others. And we'll talk about the structures of these epithelium that provide certain functions or confer certain functions. Now, the connective tissues are the ones that support, protect, and bind other tissues of your body. This includes things like bones, tendons, and ligaments, as well as cartilage. So you find cartilage at the end of bones or along your airways, and as well as fat and other soft padding tissue, or tissues that form the substrate of organs, like your liver. The structure of the liver is made of a connective tissue. And so these connective tissues are really kind of like the filler or glue material inside your body. Then when we go to study tissues, we can use microscopy. So to view these tissues under a microscope, the tissue has to be fixed. And by fixed, we're saying that it's preserved with a solvent like formaldehyde. And this basically helps to keep the molecules in place and also prevent decay and possibly even like bacterial infection of that particular tissue. Now, sections are basically thin slices of a tissue that are so thin, we're talking about on the order of micrometers thin, that these are enough to transmit light. So they're transparent. That way, under a light microscope, we can look through the tissue and then see those components of a tissue through this thin section. Now, in order to really get a good amount of contrast in the tissue, we need to apply a stain. And these stains essentially help to enhance contrast by helping to highlight certain molecules. And sometimes with these stains, we can get artifacts, which are basically distortions of the staining, where you might find like the stain kind of aggregates more in certain areas. Now, light microscopy, which is the type we're using in lab and the type that we'll see a lot of in this chapter, uses different types of colored dyes that help increase the contrast of the tissue. Otherwise, you can also use electron microscopy, and the way you get the image here is you got to coat your specimen in heavy metals. That way, when you bounce electrons off of it, you can get a nice resolution of your image.